Well,
how's your applications going? Are you managing to answer the questions and get the stuff you need?
there's, we're still going through it. Obviously, it's gonna be pushed through. But I did receive a negative answer from KTN regarding the covering of the costs of the control. And so we need to manage the size of the trials in a way that everybody, from your end can actually live through any potential losses, basically, if we're doing a controlled trial with, water only kind of thing. The other thing is,
what size does the control have to be?
Say again, please.
What size of an area does the control have to be?
Well, this is the thing. We can have two types of trials. One is where basically we're fully scientific, and the control size has to be the same size as everything else.
Yeah.
And the other one is basically something small, something as big as you can afford, kind of thing. If we, for instance, don't get the grant and we can't get Sheffield on board, we can't get NAB on board because we can't afford them yet, then there's a situation of, well, okay, well, let's do trials which we can afford. And we design it that way, and we basically try to be as scientific as possible. But for a situation where a farmer basically says, I can't, afford the size of the control, then we do something like we're doing here, where we've got a field split in two, roughly, and the biology side, the conventional side is 6 hectares, the biology side is 4.75 hectares, and the control is 0.25, and there's no replicates. So, which only means that basically, we're only learning and the farmer is learning sort of like, in general, but we're not scientific in any shape or form. So that's the only thing to keep in mind. What I'm also aiming to do is I want to go to the VC sharks and try to find investment. And if I find investment, then basically I'll be able to put into this investment that I need to cover the controls and as a cost to the farmer, basically.
So who's that you'd be looking for investment from?
Just anybody, really. Hopefully rich people. I don't want, I think rich people should should start putting their money to good use. So I know Richard knows a lot of rich people, so I'm sure he's going to just.
Far from that, Daniel. Far from that.
But no, there's I've got contacts from the previous funding round that I'll be trying to nag again and see if they're ready to put some money to that now. So we'll have to see. It's what we're trying to do is an ambitious project, and we want to. On the one hand, we want to go with the ambitious side of things and get all the answers. On the other side, we want to start. And so we want to be prepared for both. So that's why we're talking with you all the time, very openly. And, we're telling you what the situation looks like. And so, we're going to start with what we can. And we're always going to be taking steps, as we always do. I do tell people that this is the only thing that we can do always, just take steps. So sometimes these are steps back, but as long as we keep taking steps, we're going to move and get somewhere. So that's my philosophy here, or maybe Johnny Walker's, I don't know which one. Maybe Adam can comment when he actually just, logs in, one of those guys. So that's the situation. That's the short of the long. And we've got Sarah Walsh has joined us.
Can you hear me?
I can hear you.
Yep.
Yeah, sorry, no, it's my wife's her computer is Tommy Tierney.
Okay, Tommy, yes.
I was talking to you on Twitter yesterday about the IMO.
Yes, exactly. And I'm actually hoping that Adam's going to join in because he's got lots of it.
Thing's gone, so I have to download
So he's, yeah, we'll need to wait for that. Once he joins in, he's going to be able to have a eloquent conversation with you because he knows much more about these things than I do. And they have been, playing with this stuff. That's why I interjected onto on Twitter's. But he's the person to talk some details about this stuff.
Yeah, I heard a conversation. I watched one of your webinars from the 13th of October.
Mm-hmm.
The question was asked around 50 minutes in about the IMO too. So it was interesting, just trying to learn about biology. I'd say I'm doing a lot of compost as well as the Korean national farming. So I maybe kind of should be concentrate more on one than trying every little thing, but
it keeps it interesting, doesn't it? It depends.
Very interesting.
Depends of what you're trying to get to. There are people obviously who have made entire businesses around a single microbe. But what they do is they really focus their efforts on really getting to know it, and then they've got a very clear picture, and again, scientific scrutiny and all these things is included. When we are dealing with sets of microbes, oftentimes microbes that we have no clue of species yet, and this is why we want Sheffield on board. Some of you don't necessarily know the whole conversation, but obviously we've been talking to Sheffield for months now. It's just about getting the money to get them involved in this, but we just want them to be sitting on the DNA analysis device and just be running scans for us for everything, for fungi, for protozoa, for nematodes, for bacteria, and telling us what we're actually producing, and then what's surviving in the soil, and then what is actually doing the work and what's missing to do the work, so that we can answer finally the question, what do we need to put together so that we can match the yields of conventional farming. That's the aim, basically. And then the secondary aim is the climate change things where we can store carbon in the soil. That's what Richard's working on as well. So that's basically, there's a lot of work to be done, and we need money for it because we need to pay people to do that work. But getting interested in this and experimenting, absolutely. Wojtek is also the person to talk to about experimenting with everything. And I've got an updated leaderboard. I'm gonna pull it up now. Actually, hold on.
Evening, everyone. Just going back to that KNF stuff as well. Surely KNF and the soil food web stuff's pretty compatible in many ways, isn't
it?
Adam's connecting. I'm going to share my screen in the meantime. He's going to be able to answer that question, Phil. There are things that are crossovers, basically. Right. What I have done is I have updated it, and I have sorted this with a F to B ratio. And when you look at sort of the winner, we've got Wojtek's Compost being on top now in 2 places. And Niels's Cheater Compost or Cheater material is in number 3. So congratulations to Wojtek for playing in his cellar with his little worms. So well done, because this is a great result.
Yeah, thank you. Great. No nematodes, mind, but
no.
You're up there with me, Wojtek. We have the best compost in the country. Booyah.
I'm now going to try and remake it.
That's the hard part.
That's the hard part, yeah. I don't have that much. Yeah.
Yeah, basically, so now we've got 2 artists on the call, which is brilliant. This is something that is a great start. So I've been playing with this thing, and there are different results if you sort of highlight different things. So if I sort the protozoa, for instance, by column, Andrew's bioreactor is number 1, and then again Wojtek's previous result. This is not the current one. This is one you sent a few months ago. And then there's Andy's bioreactor in number 3. So it's interesting to look at it from that perspective. And if you look at sort of like the total fungi sorted that way, then Niels is crashing it because he had that material which was 14 years old. The one thing that is not on this list is Adam's, because we decided long ago that we're not going to be adding anything that we've produced. And Adam even crashes the stuff that Niels found. So I think Tom was doing. Tom was measuring Adam's Johnson-Su bioreactor. How much did you find, Tom, in Adams?
2,500.
2,500 micrograms per milliliter. So
Nice.
so yeah. So in terms of what Adam managed to get on his driveway, basically breaks the bank even on some of our concentrated microbes in his unconcentrated material. And we also found it was difficult to concentrate it as well. So that's something to think about. But yeah, this is the updated sheet now. And definitely, you know, in terms of the F2B ratio, you've got Wojtek on the top and some very good results there. So we need now Wojtek to start making more.
We need him to teach us how to do it.
Yeah, exactly.
Yeah, I don't remember what I did now. I'll replicate it.
You don't remember which recipe that was? That's no good at all.
No, I do. I had. So let me just. Shopping's just arrived, so I'll tell my wife it's here. Just give me a second. But what I did, that was a. What did I have in there? I had some cow pats, some sheep poo, some very dried sheep poo from in the bottom of the sheep shed, where you could almost see like white spores on it, where it had been, you know, it'd probably been
white spores or actinobacteria.
Wow, I don't know. It was sort of, yeah, I don't know exactly what it was, but at the
It was
time,
like it was lying sort of like below. I would be guessing that, but without the microscope, I won't be saying. Continue.
yeah, at the time I thought this is probably fungi, you know. It was, there's straw and sheep muck, and it's very dry though, very dry.
Mm-hmm.
So that went in. Then a little bit of chicken poo and straw from the bottom of the chicken shed. And then what else did I put? And then leaves, lots of oak leaves, which were fairly fresh actually. So, you know, off the trees that year. Yeah, and then sort of followed the Johnson-Su, mixed it in a cement mixer. I didn't put any wood chip in, I don't think. It was mostly leaves. Yeah. So, yeah, mixed it in the cement mixer and then poured it all out into the bucket. I probably kept it too wet to begin with, but I had it on a drip tray. And what I was doing, it's in a cellar where I've got a heating system. So it's sort of always, you know, it's not hot or warm particularly, but it's always the same temperature. And then I was actually using the leachate coming out from underneath to re-wet it. So I just kept doing that and I was sort of doing that every single day for about 4 or 5 months, and I think I overwatered it. I then let it dry out a bit more, and I think that might have helped a bit. And then I tend to just do it once a week. I just pour a little bit of water in it because it's only a small amount. So,
but even with that amount of water, because this is interesting to me, because you know I actually played with a slight variation of an experiment where I also added grass to finished compost, and then I started watering it like hell, and that was when I created a pond basically in the compost with lots and lots of ciliates. Whilst your material actually doesn't have any ciliates, and I remember when we looked at it, you know, months ago together, basically you had loads of Tachytomebae, but you had no ciliates. So it's something to sort of, you know, keep in mind there. It's, you know, you've managed to keep it, you know, still, even though you were putting that water in, you still managed to keep it in a situation which was quite aerobic.
do you remember we had those little shrimpy things?
Yes, they were especially in that leachate actually.
Yeah.
They were not necessarily in the compost, but they were in that leachate, and that's a pond, aquatic animal, for sure. They were not actually, you know, I can't remember the name now, but we looked it up at the time, and they're beneficial. They're not something to be worried about. Actually, they wouldn't probably survive in soil, but.
And I had quite a few. I'm not sure they are no, not tardigrades. They're quite big wormy things. They're microscopic. They're much bigger than a nematode. They're quite chunky.
Mm-hmm.
I can't remember what they're called now. But you know, when you first see them, you're like, "Whoa, what is that?"
Well, there could be some larvae basically, but
no, there might be a lot. I'll have to look up what it is, but I remember looking into last year. So and I think they're more wet, in a more wet environment. So I was going to say, what else? I was going to say something else, but I've lost it. I can't remember. Might come to me in a minute.
I lost it long ago. Sorry.
Yeah.
And also in terms of Vortex updates, anybody who's following Vortex on Twitters, Vortex posted a picture of a piece of pasture that he put some of his vermicompost extract, and you had an interesting resolution. I think.
I think it's well, this is what I'd like to try and do now. I think with the compost. Now I've got the compost sample results from you guys. I think I want to try and use some of that stuff. So again, I could scale it up a bit and try some field size, but I think I'm just going to carry on with the backpack and do some very set out squares in different areas after my animals have passed through and grazed, and then I can monitor that. But
I found it.
Yeah. So you could see, the video shows it better, but you can sort of see I don't know how, it's easy for me to see it, but you can sort of see, hopefully you can see a green square at the bottom there.
There doesn't seem to be any standing dead material.
Exactly. That's the point. Yeah, it really looks different in that sense, and it was full of clover, a lot of clover in there. And now there was evidence of nibbled grass blades, which would suggest to me that the deer and the geese have been on it. So that could be a reason why there's a bit less dead matter there. However, it wasn't highly grazed. It wasn't overly grazed. But what's interesting about that is I've had this before, where I did a huggle coulter experiment where I basically buried a tree in the pasture, covered it in manure, put some grass seed on, and was wondering if that patch of grass would stay greener for longer. Because as the tree decomposes, we're releasing stuff into the soil, but also hopefully keeping that bit of soil a little bit warmer and a longer growing season. And that was overly successful in that, well, the first time the animals came round, they stripped it bare. It started to recover, and then every piece of wildlife, hares, deer, everything's been on it. It's now just a totally exposed log in the middle of the field, because all of the wildlife has clearly gone to it because it was more nutritious, tasty, sugary, whatever it's attracted them. And I think
it's forage.
And I think that's what's happened here, is the wildlife has come to this spot because, the herbivores and animals are pretty clever. They like what they know where they taste good stuff. They spend most of their time trying to eat things. So I think that's what's happened there. So that to me is definitely okay. I'd totally forgotten I did this, to be fair. I did this in the spring. It did okay. It didn't see anything remarkable. And then I've come back to this sort of really probably 2 months since I've looked at it, 2, maybe 3 months, and just went past and just thought, oh yeah, that's that bit. And it just stood out, So definitely going to be trying to be experimenting with some of that, I think.
What was it you put on that, Wojtek?
That was, so I just, using a Hessian sack from my worm compost, and water, just extracted the biology and presumably also some nutrients from there, and then put it in my backpack knapsack and then sprayed it. I did also do an application, so I mixed something here, so it could be, I need to separate this out now really, because I did also do a nettle tea. So I just picked some nettles, put them in rainwater, and left them in a tub for must have been 2 months. Totally stank. And then I didn't put a lot in. I only put a small amount in with one of the applications that I did as well, because I want to experiment with nettle teas as a potential fertilizer.
And the thing is that I do believe in increasing resource efficiency with the microbes. So I think there's a thing that we need to explore more in terms of using organic amendments with the microbes and without the microbes, and things, and see if we can get a better use of them, kind of thing. So yeah, thanks. This is something that I was really happy to see when you shared that, when you sent that to me.
I can't resist an experiment. You know what I'm like. I just need to be better at sticking to one thing, and then see. I'm probably a bit inconsistent and a bit like, oh, I'll just try this now.
No, just talk to Tom. He's going to tell you exactly how to do it.
Okay.
You unmute yourself.
To do what? Sorry, I missed that.
To be exactly scientific and stick to one thing and then jump from one thing to the other.
Oh gosh.
Okay.
Could spend hours thinking about methodology. Yeah.
Yes, exactly. So yeah, so this is we've got the top of the leaderboard now, and Tommy's got a big furrowed brow on his face there, basically saying, what did I get myself into? It's like today. Because Tommy just joined us, and I promised Tommy he's gonna get some answers on KNF and natural farming with Adam, so maybe you can ask some questions.
Yeah, go ahead.
Oh, can I? Sorry, Tommy. Sorry, I remembered what I was going to. I'm sorry, I jumped up in front of you. I apologise, but it was only a quick one. With applying the biology, when do you think I should? Because should I be applying anything? I'm sort of thinking it's getting too cold to be applying stuff at the moment.
That's a good question. And so, if you do apply now, you just look at the resources that you've got, obviously, because if you put everything out now and it just basically goes dormant or dies, then you lose everything for later. But if you can, partition some of your material to also start applying now, especially the fungal component should be able to do something now. So it depends on how it's capable of after the spray, of actually getting into the soil and getting into the organic elements that it needs to survive on. But winter time is the time for fungi, really.
Yeah, because I suppose I could try something, couldn't I, anyway, because it's a small area, I could do something similar. And then the other thing was, someone said to me, how do you know it's the biology and not the nutrition coming off the vermicompost or the, and that was, I thought, yeah, that's a good question.
Boil the biology.
Hey?
Boil everything. And basically if you have two preparations.
Well, I see,
right?
So kill off the biology and then use the, right?
Yeah, so then on one hand, you've got this just wood biology, and the other hand is just the nutrients. And then you have a good comparison. And if basically the comparison goes absolutely level, then you've got your answer. And then, we learn more about this as well together.
Yeah, okay. Sorry, Tommy. Well, that's me done.
No bother. No, I had up a picture of the Imo 2 I was making on Twitter, and it just came up then about that the sugar is killing the biology. Well, I said on Twitter, it's stabilizing biology, and I've kind of looked back over where I'm getting this from, and I still see people saying it does stabilize it.
Yeah, so that's how it's taught in KNF, right? They teach that you collect IMO 1 on rice traditionally, and then once the microbes move in, you add the sugar and you massage it in. Yeah, and this apparently causes the environment to dry up, and the microbes sensing this drying, they cyst up. Now, what we've experienced here using the microscope is that doesn't really happen if you mix the sugar in really fast. And I haven't got any data on mixing the sugar in slowly, but generally when people are doing this, you're going to do it in 20 minutes, or you're going to try and get the task done pretty rapid, right? So you're going to mix the sugar in, and the osmotic pressure from the sugar we've experienced bursts the flagellates, possibly the nematodes too. So anything that's bigger and juicy is going to be exploded by the sugar. And unfortunately, KNF doesn't use a microscope. Well, Chris Trump does, but I don't know if he's got any information on this. But yeah, if you can skip IMO 2, I think you're going to have higher diversity in your IMO 3. but obviously then the inoculum is not shelf stable. It needs to be used up pretty rapidly. So you're basically using IMO 1 and you're carrying that straight through to IMO 3. And that's probably, I put money on it giving you higher diversity, particularly in flagellates and nematodes. And also you don't need to use rice, we found. We've used a mixture of hemp, buckwheat, frass even, and like rolled porridge oats, like flattened oats. So we've experimented with lots of different things. You don't need to use rice because obviously we don't produce a lot of it in our bioregion. Also, there's not a lot of information about this out there, but in KNF, they use a sushi rice and they cook it, and that's to give the right structure, right? But a lot of people in the West that come along, they start out, they'll use whatever rice and it will generally get overcooked. So that's just one thing to think about. But yeah, we've definitely experienced the sugar popping microbes. So that's one thing to be wary of. And also, if you don't have to use sugar, then why would you use it? The other thing is go make your IMO 3 pile in some kind of wooded area and do the whole of IMO 3. Just completely skip 1 and 2, go straight to IMO 3, and just rely on making that pile in the environment of a microbial rich wooded area. That would be another way to do it. And then the other way you could go about it is create the best compost you can create. Obviously, you kind of need a microscope to check the microbes. Yeah, so create that good compost and make sure it's balanced in the different groups: bacteria, fungi, protozoa, nematodes. And now use that compost as your IMO 2. So put that compost into IMO 3. And what you're going to find is that the IMO 3 process expands the populations of nematodes, sometimes very significantly. So that's well worth doing. The risk, and here's the risks with IMO, right? IMO 3 it only goes to 48 degrees. Pathogens start to get killed at 55 degrees C. So by staying under that 55 degrees C mark, we are potentially expanding mesophiles. That's the mesophilic groups of organisms. But in that group, there can be lots of pathogens. So just one thing to think about, in the IMO 3, we can actually be expanded pathogenic groups. And obviously lots of information is needed on this. This isn't the be all and end all of the process. I'm not trying to turn anyone off the KNF process, but just be wary that in that IMO 3 scenario, you can grow out pathogens. But you can also get really good numbers in nematodes and flagellates. And it's a very quick process compared to composting, right? You can get it done in 16 to 21 days, whereas generally in the UK, depending on what you're using, compost is going to take a lot longer than that. And then after the thermophilic stage, you're going to have to wait for the microbes to repopulate the pile. Whereas obviously the IMO 3 or analog using compost as the inoculum, that is going to be ready in 16 to 21 days. And it's best to use that straight away, because what we found is you have this pile full of nematodes now, but they're all competing for ecological space, right? So if you now put that IMO 3, you mix it 1-to-1 with green waste compost or something has got lots of bacteria in it, but not a lot of grazers. Now they can move out of the IMO 3 into the surrounding environment, which is rich in bacteria, but doesn't have lots of grazers like nematodes. And that gives them more ecological space to spread out and keep breeding. And you avoid a population crash where the population crashes because of lack of resources. So that might have been a bit of a mouthful. I hope I didn't overload you, but if you have any questions.
The problem I found with the KNF is I have a microscope and I've been looking at it for a few years, because I have a vermi bin and I'm trying to make compost, but I could never really find what the KNF should look like.
Right. Yeah.
See exactly what I should be looking at in KNF.
Yeah, 100%. And are these molds we're seeing, are they beneficial to the system. And I think this is really dependent on the system. You know, if you're growing macadamia nuts like Chris Trump and you have a rich mulch layer, then maybe some of these early successional decomposers are going to speed up decomposition and lead to general well-being in the system. But if you're applying it to, say, a grain crop that's in an ag field that's very degraded and there's not much else around, then there's not much else to compete with those pathogens or potential pathogens, maybe we could see a problem in this environment. But again, more data is needed. People do use these systems to grow stuff and they are successful. They don't really use them on grain as far as I've seen, apart from rice.
Yeah, and rice is also it's a crop that basically can survive anaerobic conditions. And so if you think about basically some of the microbes that we see from the KNF side, which kind of is the opposite of what Dr. Ingham talks about in aerobic systems, they kind of might be going hand in hand, because there's a reason for the fact that that rice is grown in paddies. It survives the paddies, it survives the anaerobic conditions, whilst weeds don't. And so that's why they grow them in paddies. But then that makes them maybe a good partner for some of these microbes grown with the KNF systems. So it's something to keep in mind. This is why we are seeking grants and investment to do lots and lots of trials, because we really want to nail down a microbiological system that's going to work for the UK, and that's going to be the end all be all, basically. So
a few more notes on the IMO process that we found that work really well. They generally use for IMO3 a carbon source and then a carbohydrate, which in the soil food web community we would refer to as a high nitrogen input. If that high nitrogen input is of an alkaline nature, there seems to be more oomycetes pop up in the pile, right? But if you pick something that's acidic, like coffee grounds, that drives the pile into an acidic ecological scene, and that wakes up maybe more stuff that's beneficial, like beneficial fungi, flagellates, and it impedes the growth of oomycetes or pathogenic fungi that would most likely grow in a more alkaline condition, a reduced condition. It's really interesting to follow the work of Olivia Husson on soil redox potential alongside the KNF stuff, because it's helping me drive the piles into an ecological scene that looks a lot more beneficial for plants.
Cool. Wojtek?
Yeah, hi, sorry. Yeah, I was just remembered that on those compost ones, they were quite small bins, so they didn't go thermophilic actually, which was something I was worried about at the time. But I don't know. I suppose what I don't know is whether the fungi in there is potentially pathogenic, or I suppose you'd see that.
No, because yeah. Look as done a proper count. So it's not the thing with yours is, didn't you have worms in it in those JSs?
Yeah, I have.
But the IMO3 doesn't.
Sorry.
But the IMO3 doesn't.
Yeah. Okay.
So basically, you also had an element that works against pathogens as well.
Yeah. Okay. Yeah. Well, I do think doing stuff on the woodland floor is a great idea. I think putting your composts, in that sort of environment.
Yeah, especially next to your miscanthus piles.
Yeah, exactly. Yeah, rotting miscanthus for 5 years is good. Yeah. Still haven't worked out what those little crystals are, though.
Yeah, I mean, there's some of these things in that.
Yeah.
I definitely would take the idea, maybe of trying to go into because I don't know, there's something nice about being in the wood. It's an old oak trees, and there's plenty of fungi on the ground. And
we had lots of good results with oak materials.
We're conventional farmers trying to reduce pesticides and synthetics, so it's actually lovely to get into a wood and get away from that. So it would be nice to be actually doing something in this wood you know, maybe not the IMO so I was doing, but maybe bringing a bit of compost to the floor of the.
It would bring more necessity to conservation of these areas around farmland.
Exactly, I agree with Adam. You know, this is something that you know, if you started actually doing something for growing out the beneficial microbes, you know, in the woodland, and other farmers would start picking it up. You know, maybe we would have a more sort of like a internal incentive for.
Right, we can skip the rice, we can skip the sugar, we go straight to the source of the microbes with all the materials that they eat.
Yeah,
It's where all the biology is hiding, isn't it, in the woods?
yeah, you know. The thing is that there's a lot of work, you know, like on HDB and or NIAB side, you know, about feeding microbes that are already in the soil, but the soil bank is not really as diverse as a healthy woodland bank. And so if we were to help the woodland bank grow, and then take that and start reapplying that to agricultural soils, I think we've got a bigger chance of helping the whole thing.
Another super interesting thing is sometimes maybe pathogenic fungi are good when your target is an aphid. So we could, you know, fungi that eat chitinous material, the exoskeletons of insects, if they could be cultured, they could be used with agricultural systems to protect the crops from pests. So it's all about context as well. It's not like pathogens are bad. It's all about the context in which we put them to work. And you might be able to culture them by including frass or chitinous material in the collection material, in the pile that you turn that's in the woodland, because the woodland no doubt has lots of fungi that degrade chitin. So there's my little tip for the day.
Well, no, that's what I can. That's one thing I definitely take from it. Anyway, the other, you were on. Someone was on there earlier about the Nettle Tea, and I actually had been kind of doing that as well, but I had been doing it in the Korean national farming type of with Japanese knotweed, lucerne, and nettles. But I had been making a FPJ, so I'd been using the brown sugar again.
Yeah, but in that case, the sugar is used to draw the sap out of the plants and keep it shelf stable. So yes, it's going to be taking out some microbes possibly that are on the surface of the plant, but you're still capturing those growth hormones and the saps, and then you're making that shelf stable. And as that ages, I've witnessed using the microscope, the yeasts do pop up in there, and some bacteria are still alive in there. And it all depends on whether you go to that super saturation level, where it's, you know, if you've got a kilogram of material you're extracting, if it gets to a kilogram of sugar with that, you're gonna really bind up all the water. Every water molecule in the ferment is gonna be bound with a sugar molecule, and that's gonna make that environment really dry. But if you don't hit 1-to-1, then it's more watery, more fermentation is possible still, and microbes can still propagate. So there's a lot of work to be done on the ratios involved in making the FPJs, but I still think FPJs are a really good thing, as well as anaerobic fermentations of nettles, comfrey, anything else, as long as they're handled in the correct way. Like, for instance, we would advise from the soil food web lens that if you are going to make a nettle, comfrey, whatever anaerobic tea, you could put it in a bubbler for a day or two to try and bubble off any alcohols that are made in that process that aren't necessarily going to be good for the plant, but you're still going to hold a lot of energy in that brew that is good for the plant, right? So we're just trying to mitigate anything bad that can happen from using these processes through the lens of the soil food web, which I think is the right lens, because it's the people really using the microscope to bring accountability to these processes.
Yeah, and just like lastly, you know, about the use of sugar and microbes. The thing is that, you know, it's worth noting that honey contains bacteria. It's just that they're basically, you know, they're not something that that can develop because of the concentration of the sugars in honey and all the enzymes that it contains. But it does contain bacteria, and this is why honey is not fed to infants, because they don't have the the diversity of microbes in their gut to be able to fight off, was it bacteria, the ones that cause botulism, right?
Botulism, yeah.
Yeah. So the thing is that, some bacteria will survive that process. What we have seen, the reason we speak from it, we always speak from experience, and what we have seen is that the higher level guys, they just get burst, and these are the guys that give you nutrient cycling. So, I see these preparations basically as a nutrient preparation more than a microbial inoculant. The one positive about these nutrient preparations, when you're doing that in a fermentation process, is that microbes have a knack of gathering everything and giving you a balance, basically, of these nutrients. And I think this is something that that is the main advantage of these organic preparations, that you're not just simply sending out a single nitrogen synthetic feed. You've really have got a balance of nutrients, and this is what makes it work. But again, in terms of microbes, it's going to be limited.
An interesting one we found as well is if we go to our local farm shop, they generally produce a lot of waste because they're a pretty farm shop, and if a carrot has a little mark on it, they can't put it on the display, so it just goes into the skip outside. So we've collected stuff like potatoes, carrots, turnips, parsnips, and these are all root vegetables storing carbohydrates that are very much like plant exudates. So now if you take these roots and you put them into the FPJ style process, you chop them up and you mix them with sugar, you can draw out that carbohydrate, that plant the exudate like compound into the sugar across the cell wall in a cold process that keeps it relatively intact. And then if you use that up front in a compost tea or a protozoa infusion type situation, then it seems to be really good for flagellates. So that's worth thinking about. You can, if you're going to buy molasses, say I buy a big 25 kilogram bag of molasses from Billington's, and if you want to make that go further, you can just mix it with the whatever plants you've got, whether it's fruits, leaves, but these roots seem to be really good too. Any kind of stem or crown kind of vegetable, like a carrot, turnip, beetroot, yeah, any of these things.
Yeah.
And if you ever see a big pile of sugar beet going left unused because it was rejected or something, perfect candidate for extracting the exudates using the FBJ or fermented plant juice type process.
Perfect. Thank you very much.
No worries. If you ever got any other questions, you're welcome to email me, Tommy, because I might be able to help.
Yeah, I will do, definitely. So thanks.
It's just adam@soilecologylab.co.uk. Cool. And then Judith, you wanted some advice on actually building compost, right, yourself?
Yeah, I need to get going on.
Are you up for turning, or do you want to go in for a static pile?
I've said I'm going to do a small Johnson Sioux.
Yeah.
So I'm kind of gathering stuff up for that at the moment. But we've got 70 odd cattle, store cattle in on straw. So I think we should be doing something with that dung.
Yeah.
I need to work out what best to do with that and what I need to add to it to make good compost. And then I guess that'll need turned because there's going to be too much of it to do.
Depending on ratios, yeah. So if you figure out,
sorry, before you start, actually look at the material first, see if there's worms in it already, because you might actually have worms in it already. Because they love this material. The actual composting worms actually, live in cattle already. And so if you have the worms there, it might be the case of just what Adam started probably talking about in terms of ratios of how much nitrogen-rich materials versus carbon.
Figure out how much bedding is actually in it, and then how much manure is in it, and then you can kind of take that, and how much manure is in it is kind of your high nitrogen input, and then you can include the bedding as part of the carbon input, and then
add more.
yeah, so you could have like say 1 part high-end, 3 parts green material, and then 6 parts carbon or something like that.
So we have been working usually on the assumption based on observation that the bedding that we've used here was 70% high nitrogen, 30% carbon.
Right, and that's from, is that, you know, like from a cattle shed or, yeah?
Yeah, beef boys.
Overwintering.
Yeah. So very, very deep litter, basically. No, not something that, you know, Adam had some better ideas for deep litter from actually in Korea as well. But in terms of the, you know, when they empty it, it's going to be 2.5 feet deep at least.
Yeah, that's kind of where we are, I suppose. Yeah. Yeah. And to that, you need to add green material and more carbon. Is that what you're saying?
Yes, because the thing is, you know, with just the bedding, it quickly goes anaerobic. It basically starts sort of heating up in pockets. You have to turn it, and it's a difficult material to manage. I mean, it's best to if you got lots of it, obviously you need some machines really to play around with it.
Yeah.
So yeah, you know, it's best to have some other material, whether it's just straw or having some leaves, you know, some other wood chips for carbon. In terms of the green material, can be anything from fresh grass to hay and silage. In terms of sort of like a larger, bulkier amounts of material available on farm. You know, obviously you're
any rejected silage, any silage that doesn't meet the cut for the cows.
Exactly. You can use stuff which is, you know, something that cows, you know, shouldn't be eating, but you can still use it as a green material.
Yeah.
But that's gonna be a hot compost pile that will need managing, obviously. So we'll need active turning.
Yeah, you need to monitor it and turn it at the right points, right times.
Yes, I mean, yes, you do. There's a, you know, if you for instance, you know, don't necessarily always, it depends also on sort of like the size of it, but you know, Johnson Soup bioreactors work on the basis that they've got a constant access to oxygen.
Yeah.
And so, you know, if you go on the Johnson Soup bioreactor groups on Facebook, there's one very active so worldwide group, for instance. There are people that have gone windrow size, where they just put a massive amount of these tubes when they first load when they first build the windrow, and then after a day they pull them out, and you know, they've got like a sort of a Johnson Soup windrow kind of thing.
And I mean, when I look at that, do they have air coming down from beneath, or does that work that the air can just go up and down the one tube?
That's a good question.
Because to me, I kind of feel you want actual circulation, so it comes in from the bottom and up the top.
That's a good question, and the ones that are more diligent basically have a layer of pallets at the bottom, so they still work on the premise that they're going to have access to air from the bottom.
Right.
But yeah, it's a very, you know, it's one way of doing that. Another way is poking lots of holes on a regular basis, and that alleviates the you know, the low oxygen, because the pile, you know, it gets hot, and even when you poke the holes, those are just going to start venting, you know, because steam's just going to start coming off of it, and that will create pull, and that air will have to come from somewhere. So the pile will start pulling from, you know, other parts of it to compensate. So there will be some draw of air, not as perfect as having a column like in a, you know, proper Johnson Soup. But I'm just basically thinking of ways to limit the amount of work for you if you're working with a, you know, large amount of material, and what sort of equipment have you got without a turner, you know, because the ideal is to have a turner and somebody monitoring it all the time, and that's basically the, you know, the ideal. Not everybody has got the luxury. I was speaking to a couple of guys from a company that grows salads, and you know, they don't complain on resources. They've got a turner, they've got everything to deal with it. So, but not everybody's got that luxury.
Are they easy got hold of? I mean, I can't imagine that. I mean, I've never seen one actually advertised for sale or how much they might be?
I know there's farmers with turners in Scotland because I've seen them.
Right.
So
Okay.
if you maybe ask around, maybe there's somebody there, and they will have PTO driven turners even.
We built one last year from out of a trailed moor. If you go back through my Twitter, I think I have videos of it. We converted a moor and we did it all in for about €3,000. And you know, we're not engineers, but we were able to do it ourselves on farm, and it's working fine. Because I know one of my friends imported one from Scotland, a 10-year-old one for €8,000, whereas we built one fairly straightforward, and there's videos of it on my Twitter. You know, just a converted silage moor turned into, that's what we're using, and it wasn't, you know, we just did it on farm, and there's videos on my Twitter if you want to see it, but that's what we're using now. So,
Right.
If you find them, Tommy, and email me the links to those. I'll forward it to Judith if you don't mind.
yeah, no matter.
So at least you can have a look at it.
And then another thing, Judith, that I was thinking about is for greens, if you have access to any understory moss. I know Scotland's, you know, moss is everywhere. So moss we found is a really good substance for holding air and structure. Yeah, it's very resistant to the composting process. So even after 5 heating events in the pile we tried, it has moss in there. It still looks kind of morphologically the same. It's a bit cooked, but it's holding everything together. It's providing a long-term green, and it's, you know, if you've got it in abundance anywhere local to you, then it's really good.
I've got my brother-in-law does a lot of scarifying. I don't know if you know, but I mean,
yeah, they do it on the lawns here in the South Downs because they get very mossy.
I can't imagine. Yeah, I mean, we have 60 inches of rain a year, although I don't think we're anywhere near it yet. But we've got 5 weeks to make up that average yet.
It's going to come all at once.
It could do. It could do. It did it last year. The last couple of months last year, just, yeah, it was unbelievable. But yeah, right. Okay, no, that's great. Lots to think about and lots to have a go at. Yeah. I quite like Wojtek's idea of doing trial squares in different paddocks and things. That's my kind of stuff. Yeah.
Yeah, it doesn't have to be big, right?
And see if you do that and it works. Does that biology, you know, if cows are eating that and the biology is good, does it get spread out elsewhere?
It doesn't really do it that well. It's, this is it's not just, you know, what we see, but this is something that comes from other places. They need taxi cabs for that. So, you know, biology oftentimes will be carried by animals, but we're the main taxi cabs these days. So it's we have to apply it, and then it stays there.
You actually need to put it on every square meter kind of thing to get it there.
Yeah, you kind of have to. It's not
some of it's going to spread out,
but not terribly, you know, easily. You know, we've heard basically of people, yeah, eventually, but you know, nature doesn't necessarily work on the same time frames as we do. So,
right, after 2 or 3 years of applying it, there will be a background level that's gonna sit in the field, and yeah, that's gonna just grow naturally, but within the first couple of years, probably not.
Yeah, but this is why I have these discussions with people who tend to think that, oh, you know, it's like, as long as you stop applying, you know, chemicals, the biology will come back. You know, you're gonna have your cover crops, the biology will come back. And I said, from what? Hedges? They really don't have a bus transit system. You know, they don't travel that quickly. And you know, we've had samples that people have put in herbal lays and whatnot, and they've been sending us samples, you know, to see what's going on over months. Absolutely zero change. You know, so there's the thing is that we have to apply them, and once we apply them, suddenly 2 weeks later there's a population in there because they actually had the bacteria to grow on. So it is a mixture. We have to have a good inoculum, and then we need to support them when they're there. So
yeah. which to me is why when people try to go organic without doing any extra stuff to it, it can be a disaster, because
yeah, we've seen this with our.
We're kind of talking about going organic in that way, but actually facilitating missing out that 10-year gap when it could be awful.
Yes, this is why we exist. This is what we want to solve here. So because it's like this is the thing that frustrates me. It's like I've spoken with farmers. Yeah, I can't go organic because I'm going to have a yield penalty. We look at the market. Yeah, organic bread costs £4 a loaf to £12 a loaf. We speak with the grain merchants. There's no growing demand for organic grain. Well, of course, because people can't buy £4 loaf of bread. So who's going to move first? So yeah, so that's why we want to try and solve it.
Mm-hmm. Yeah.
It's kind of like the difference between organic and regenerative. Organic is sort of just the absence of certain chemicals and fertilizers, whereas regenerative, there's sort of an active input of biology, kind of making an effort to bring back biology faster than it would just occur naturally. There is
a farmer in Scotland that I was chatting with, who's been organic for a very long time now, and because of the kind of rotations that they do get 5 tons of barley per hectare, which is something that is a good yield for a conventional farmer as a low input barley. But then he doesn't grow it every year. He grows pulses, beans and peas and all these things, throughout the other years to be able to replenish the nutrition that he's lost in just one season of growing barley, for instance. And so if you're talking about having the same barley yield that we're having right now, where there's 3 million hectares grown with, 1.5 million hectares grown every year in this country, then it's not going to happen. We're going to have 1/5 of that grown every year to be able to compensate. So if we want to close that yield gap, to go organic, in the sense that all the conventional farmers that are growing barley and wheat every year, they can still actually grow wheat and barley every year. We need to do something differently.
Just one question. So, like, I have brewers built. I'm using them for a few things, but like, I could still stay with the same system of, I have a large worm bin too, of trying to do a compost extract the same way with what I produce in the floor of the forest, maybe mix it with what I have in the worm bin and still go with the same system, Ingham's extract, like.
In fact, I think that's better. We rarely achieve all the groups in one pile. So here we generally take from different piles to achieve a balanced inoculum. So you're right on the money with us. Yeah, that's what we do too.
Yeah, because when I've been dressing seed with biology up in Arger, I've been taking bits of everything, like a bit of lactobacillus bit from the worm bin. Yeah, there'd be 5 or 6 different things going on. And the same when I was doing soil drench, I do soil drenches onto cover crops this time of year. I've been bits of everything, and we're still, where I'm going with that is okay anyway, isn't it?
How do you do your dressings? Do you put some compost in the hopper ever?
And where I got the idea is that video of, did you ever hear of Ian and Di Haggerty in Western Australia? Christine Jones would have a lot of dealing with him. They put a video, you just simple an auger and let it free flow from a tank into the auger as the grain is going up the auger into the
fluid?
Yeah. It is a fluid, a liquid I'm putting onto the seed, we'll say.
Yeah, but this is like inside the hopper, basically, you mean?
No.
Yeah, just at the very start. I have a pipe at the very start of the auger. The grain goes in the hopper and goes up the pipe, and the liquid is flowing in at the bottom. So it's mixing with the seed going up.
Just basically as the seed is coming out, basically there's a
No, as it goes into the auger, not comes out of. So all the way up the auger, it's getting mixed up with the fluid.
Mixing up.
All the way up the length of the auger. Yeah.
This is where basically my lack of understanding of farming machinery basically makes it.
I have another video of that on Twitter. I'll send.
Okay, I'll have a look at your Twitter account. Might be able to understand it better because
Yeah, it sounds like a really good plan.
We're trying to solve, this thing application for, what's going to be the best way for farmers, the cheapest way to modify equipment if they need to. So.
And does that work for you, Tommy?
Pardon?
Does that work for you? Do you see a difference in the crop? Have you done a bit without it and a bit with it?
Yeah, that's of all, and I've a lot of stuff done in the last 3 years, stuff built to try and do, but that seems to be the best. Like we adapted our no-till drill to put down liquid and extract, but we just had more problems bringing IBCs following us around and blockages, and it was slowing things down. But then dressing the seed, it's uniform on the seed. It's simple. It's only a bit of gravity, and it just seems to be working. It's nearly something we've done on the farm in the last 3 years that works the best.
Are you just putting that through a sprayer nozzle as you're putting it on just before it goes up the auger? Is that through a sprayer nozzle, or are you just it's just opening as if it's a tap, tap running on hose?
Yeah, a lever. It's a 60-litre drum. We just put a pipe off it with a valve, and nothing scientific. I just turn the valve a quarter turn and let it flow down, and I go into the hopper, and I just kind of know roughly, the way it's flowing in the hopper where I have enough on.
Yeah. So that's as you load in the hopper. So it's going up the auger into the hopper, and you're drilling it within an hour or so. Yeah.
Going in, like we were looking at doing it the day before, but I had it in my head. I'd like it going in fresh.
Yeah.
And like obviously we have to slow down the drill a small bit because it's that bit damper in it, but it's not bad. We can still get good acres done, and we've kind of tweaked enough now that it just works very well.
What are you drilling with? What direct drill are you using?
The John Deere 750A.
Is it? Yeah. Yeah, because that's I was wondering about putting some liquid, because we've got a 750A as well, a liquid for putting in the seed slot, but it's probably easier just dressing the seed then.
Yeah, because first thing we did was adapted the drill to put liquid down the slot, and we used Clive Bailey had pipes developed.
Yeah, it's nuts, isn't it? Yeah.
We used all that, and at first we were just using liquids we were buying, but then when we were trying to make the extract and our own bits and bobs, we just found there's something always blocking. The pump wasn't running kind of right, and then you are bringing IBC tanks with the drill the whole time and filling them up and unloading, and just when you're under pressure, and in Ireland windows to plant are fairly tight, so you need to get a lot done. At the one time we just found seed dressing just seems to work very well.
Yeah, okay, cool. And obviously it's the grain's still running pretty freely. It's just getting a dose of seed dressing on it. Well, biology seed dressing, and it's not wetting it through by anything. It's not like a soak. It's just wetting the surface, huh?
Yeah, it's wetting the surface so that you're hoping the biology you have in suspension is now on the seed. The seed would look damp in the hopper, but not bad enough that it sticks in the hopper. It will still flow away. We've kind of got the balance right.
Yeah.
We've learned a few hard mistakes where we've get in and push it all out on the ground. But it's just a bit of
trial and error. Yeah.
Yeah, and of everything we do, that seems to be working the best, because we can get a bit of every type of biology I'm doing, mix it together and onto the seed. And I have done things in the greenhouse. My mother has a greenhouse, and I have been, trials to see what works best on the seed.
Cool.
I just find, that definitely.
What's your Twitter handle, Tommy, if you mind me asking?
Sorry, now I'm not great on Twitter. I actually don't know what it is, but
hold on, I'll find it.
Thank you, Daniel. Thanks, Tommy.
Yeah,
that's interesting.
I'll send you on the video of the dressing the seed and the compost turner we built.
It's Tierney
1981.
You can see it, yeah?
Yeah.
Okay.
Ideal. Thank you.
I've got it. So yeah, we got talking because of this. And
because a farmer I know, a local farmer, went over to Chris Trump, and he looked at that and he said, in Ireland, you should only have yellow, blue, green, and white. And he said, you've red. He said, you overcooked the rice or you left it too long. And I said, I did. I left it for 2 weeks. And he said, no, 5 to 7 days max. So he actually told me, throw it out. And I will, like, look, it isn't as if I'm after investing a lot of time and money in doing it, you know, but he knew straight away. He said that was either overcooked rice or I left it there too long. And I said, yeah, I left it there too long. And that's why you've red in it. But yeah.
I wish some of this advice came with any accountability on what those colors actually mean.
Yeah.
Because yeah.
That would be great.
I mean, I've been heavily studying KNF for a while, and I'd love for it all to be true, but some of it's just a bit like, I don't know how people look at colors of molds and they know that, like, what hyotrophic grazing organisms are in there. I just don't. It doesn't cut it for me. But who knows?
It's like, you know, it's like with everything, you know, this. I mean, even you, Adam, you found a paper recently on some biodynamic preparations, and biodynamic preparations, you know, have for the longest time been, you know, based on experience, and there was a lot of questioning around it. And years ago, I have, for instance, found that somebody looked at biology of some of those preparations, and then you found papers on some substances and you know the quantities that you need for the substances, you know, the minuscule quantities that you need these substances to do anything. So the thing is that there's truth in, I'm sure, you know, that the actual validity behind these methods lies in, you know, in experience of the people who've created them. But then if we want to recreate them, you know, I think there's going to be a need for some more scientific scrutiny to be able to really answer these questions. So it's an interesting subject. It's the rabbit hole that we keep going down. I think Doug's got an unanswered question though.
Yeah. Okay. So I'm always looking to see how we can. I was looking for the mind blown emoji a minute ago on the chat, but couldn't find it. I always find these conversations quite amazing. One of you mentioned, you know, a well-balanced inoculant. So, okay, let's say winter wheat then. What is a well-balanced? What should be in a well-balanced inoculant? What would you like to see in a well-balanced inoculant?
So there's a you know, we're now basing this on Dr. Ingham's work, where basically she looked at different ecological niches, and the ecological niche of wheat is the same as sort of a kind of a meadow, kind of a pasture kind of situation. And she finds that there is a balance between fungi and bacteria in terms of their biomass. And so we're looking at a balanced biomass of fungi and bacteria in the soil for any of our row crops, roughly speaking. They usually have a range. It's not usually just, you know, 1-to-1 has to be, no, they usually have a range. And so it depends on the plant. But in terms of winter wheat, in terms of barley, in terms of corn and pastures, it's 1-to-1 fungal to bacterial biomass ratio. And then we're looking at sort of minimums of biomass. And in the growing season, you know, I think if I'm speaking from memory, I need to pull it up. So I'm going to say, okay, well, minimums is 135 micrograms per milliliter of soil of each that you want. That's like the minimum, but you want to have more, you know, 500, 600. Usually you have more of bacteria. They usually start at 1,000, 1,500 micrograms per milliliter of soil in soils which are very, very overused. We've seen 6,000 and more. But the thing is that you want it balanced for these crops, and then you want micrograzers. So you want the protozoa and you want the nematodes. So again, Dr. Ingham advises at a minimum of about 100 nematodes per milliliter of soil and about 10,000 of some protozoa in the soil per milliliter is required to get that nutrient cycling. So we always try to aim higher with these things because, you know, the more nutrient cycling, the better. The way it works basically is that, you know, even if they pee out some of the nutrients and the plant for some reason doesn't use it, there's a bacteria that's going to pick it up, and then the bacteria is going to get eaten again by the micrograzers. So this nutrient stays in the system for quite a while. It doesn't leach very easily. And because everything happens around the roots, because the plant is feeding the system, then everything is available to the plant. So those are the minimum numbers that we'll be looking at to get the system going. At the moment, we've got on the winter wheat field that we're working with, we're currently still in a situation which is very much bacterial. We now have bacterial feeding nematodes established, but we've only now seen an uptick in fungi. So we've got, I think Luca was looking at it, and he measured about 0.14 fungal to bacterial biomass ratio. So we still have a ways to go with that field. So there's still loads of bacteria to you know, in that field. So there's you know, it's quite a bit of work to get there. These are the numbers, and to be able to get to that balanced system is going to be a bit of work to do that.
And in a practical situation, does it take 2, 3, 4 applications of microbiology?
That's a good question.
On the field and the conditions in the field and how you can actually, because
yeah, we haven't really worked with pastry, yeah.
It's like nudging it from one to the next and nudging it again and nudging it again to get it to where you want it to be.
Yeah, I mean, you know, the thing is that there's still all these unknowns that we want to figure out. No, I don't think you can overdo it. I think this is something that's going to be difficult to overdo. The thing is that, you know, as farmers, you don't have the luxury of very, very, very high quantities of this material. And so it is going to be a case of, you know, applying what you can apply and then self-measuring 2 weeks later and saying, okay, I still need more, and sort of applying again and then again until you get to the point where you've got it. Where, you know, the concentrate of biology that we're launching to gardeners basically is on the basis that they can take the 200 mil and put it in a 1 cubic meter of their growing medium and they're done. They've got it. So that's the sort of thing that, you know, that if you have that sort of application rate, then, you know, you would pretty much solve the problem straight away. But then that application rate basically translates to what, I don't know, 2,000 liters of extract of concentrated extract per hectare. That's not even feasible, you know, with what we produce, because we're not producing it at scale. It's just, you know, it's just too much. Nobody's producing it at scale. So we have to basically look at smaller, you know, inoculation rates, and then try to maximize the conditions for these microbes to survive the application and develop. So there's that challenge. But yeah, it's not something that we can yet do in one go. But we want to do more studies so we can tell you, it's like, yeah, if we basically do this, and that, they're going to establish straight away. If we get this amount of material straight away, and we can do it in one go. But we're not there yet.
If you had unlimited resources in terms of money and access to a whole load of input materials, and you had the space to make huge amounts of compost, you could probably do it in one application. But even then, it's kind of like an unknown. You're making a lot of going out on a limb, guessing that that's the amount that this field needs, whereas multiple applications, you get to track it over time, see how those microbes adapt to that particular niche, that microclimate of your field, the levels of nutrients available, and the plants that you're growing. If you're to do it with just one application, it's sort of, we were joking that even unlimited resources, it'd be hard to do it in one application. And so multiple applications seems to be the way to do it. And
I think
these
it's not even,
higher.
I think it's not even the case of just money, but it's a case of time, and these systems have to balance themselves out as well. So there's that too. So yeah, it's a big subject. We've got a lot to do. We're already 15 minutes over time, so we're taking too much of your time as usual. But thanks, Tommy, for joining us today. It's really great. It seems that I would love to.
Yeah, thank you, Tommy. That's good to know about your seed drill.
I would love to know. I'll have a look actually at your Twitter, because I'd love to actually know how you're.
I sent them onto you there, the 3 videos onto your Twitter by Messenger on Twitter.
Cool.
So you have them addressed.
Awesome.
Whereabouts in Ireland are you, Tommy?
Tipperary.
All right, yeah. Nice.
South.
My sister's in Waterford, and her husband's family are in a farm near Portlaoise.
Waterford's only over the mountain from me.
Yeah.
Doug, to answer your question, just one last question to answer. How much does it vary between crops? Brassicas don't need any fungi. So if you're doing all seed rape as a break crop between barley and wheat, like most people do these days, it's you're sort of going back in succession and going forward in succession, back and forth, and this is not good. And so it's something that you start off with no fungi on the brassica end, and then as long as you have some good nutrient cycling and a bit of fungi, all your vegetables are fine. And then you need sort of like that balance for row crops and pastures. And then if you want to go into fruit and trees, then you really need to bump that fungal component. So I'm really, really pleased that we now have this group with lots of fungi, because now actually, I can go to James, from Luddington, and say like, yeah, we can do some trials. So we're preparing small trials with fruit now, with apples. So Gala's going to be grown with microbes. Right, guys. Thank you very much for coming again.
Smashing. Thank you so much.
Thank you all.
Everybody. Yeah.
Good chat today. I really enjoyed it. Thank you.
Yeah.
Thank you, everyone.